Hey, James. Good to see you.
Good to be here.
Like the shirt? Like the shirt.
Thank you. I saw you wearing it maybe it's a class or two ago.
A couple of weeks ago, or maybe it was last week. Yeah. All right. We got 1, 2, 3, 4, 5, 6, 7. So we're missing half the class. Let's wait a little bit. And just real quick, while we're waiting for people, where is it? Here's the paper we're going to be reading: Intracellular Distribution of Dihydropyridine Receptors. And let's see if I can find my. Can't find it. I can't find it. Okay, stop the share for a second. Okay, this is what I want. Okay, everybody, if you haven't yet, please download the paper that we're going to be reading, which is right here in Moodle, DHP Binding Intracellular Redistribution. If you don't have it, you should pull it up on your computer, although I will be showing mine. A little bit worried about this particular exercise. I think it's really important because at this, I know I said this Tuesday, but I have to fill time, so I'm going to say it again. At this point in your career, you should start reading some of these papers. And reading these papers is a bit of a challenge at first. The big thing you need to get over is getting stuck on a word. If you don't know a word, look it up real quick, get back to it. And, you know, a really complicated paper could take a minute or two per sentence to get through until you get good at it. So we're going to do a little bit of reading here. What time is it? 9:34. 1, 2, 3, 4, 5, 6, 7, 8. 8. How many people are in this class? I think 12 or so. Well, the cool people showed up, and that's all that matters. Okay, so why don't we go ahead and get started here? I'm going to do the reading, but I'm going to be asking questions. Now, no one ever answers any questions, so I'm going to have to. No one volunteers to answer questions, so I'm going to pick on people as we go through this. So be aware, especially you quiet people. I'm looking at you, Bezuit. Be aware you're going to get picked on. Okay, so let's start here in the introduction. So I think this is a good introduction for, there she is, Bezuit. Hi, Bezuit. So this paper, this introduction is good because this is exactly what we talked about on Tuesday, the inotropy in the heart. So can you guys hear the noise in the background of me? Can you hear this background noise? There's cars going by. No? Okay. Okay, I'm going to read. In the heart, a very low calcium concentration between 10 to the minus 5 and 10 to the minus 7 molar is a prerequisite for the proper excitability and contractility of the cardiac muscle. And furthermore, the intracellular calcium concentration oscillates during the systole and diastole. So systole is contraction, diastole is relaxation. The very low intracellular calcium concentration is maintained by a concerted action of dihydropyridine receptor, and we just call this the voltage-dependent calcium channel. So I did tell you this last time, that the dihydropyridine is a drug that binds to it. So the channel at the surface, calcium ATPase at the plasma membrane, and sodium calcium exchanger at the sarcolemmal membrane. The oscillation of calcium concentration during the systole and diastole is regulated through calcium ATPase, the SERCA on the SR, ryanodine receptor on the SR, calcium-induced calcium release, and phospholamban in the sarcoplasmic reticulum. Sorry about that. The progress in the molecular pathogenesis of cardiac disease, such as the ischemia reperfusion injury. Okay, you're up first, James Faraci. What is ischemia and reperfusion? Do you know what that means?
Ischemia is the loss of blood flow to something, and reperfusion, I'm guessing that it's when the blood flow returns after.
Right, exactly. So when an artery to the heart, coronary artery gets blocked, that is not totally blocked, but really blocked a lot. That's ischemia. Not enough blood goes there. And then reperfusion is when you open that block. So things change when the cell gets hypoxic, low oxygen, and then when the oxygen suddenly comes in, boom, it causes a whole lot more damage. So that's a big big source of pathogenesis there. Ischemia, perfusion, hypertrophic cardiomyopathy. Let me change my lighting because I can see my face and I know I'm prettier than this. Hang on. Wow, that's terrible. Why is this so bad? Oh, that's worse. Nice. Right. I think it's just the way the camera's picking me up. All right, forget about it. Hypertrophic cardiomyopathy, Mike West. Hypertrophy, what does that mean? Hypertrophy.
That is evading me at the moment, but I'm gonna guess that cardiomyopathy has something to do with it, like contracting improperly.
Well, pathy is always sickness. Myopathy, sickness of the muscle. Cardiomyopathy,
The myosin.
right? Well, no, of the cardiac muscle in general. So cardiomyopathy is sickness of the muscle of the heart. Hyper is over. Trophic means growth, so it's overgrowth.
Okay.
So hypertrophic cardiomyopathy means the heart grows too big.
It only has a certain amount of space it needs.
I looked it up earlier, and it says it's the thickening of a part of the heart.
Right, exactly. And we'll talk about this later, but basically if you've got high blood pressure for a long period of time, the muscle gets thicker because it has to deal with more pressure, and then it doesn't function as well. So hypertrophic cardiomyopathy and heart failure. Heart failure is when the muscle loses its strength and it can't contract as much. All right, let me read that again. Progress in the molecular pathogenesis of cardiac diseases such as ischemia, reperfusion injury, hypertrophic cardiomyopathy, and heart failure have provided a cause and effect link between the altered dynamics of calcium transport proteins and myocardial dysfunction. Okay, so this cause and effect link between the myopathy and dynamics of calcium transport. So calcium is so important in the heart. Previous studies from this laboratory have indicated that the dynamics of a number of cardiac regulatory proteins, including the SR calcium ATPase, the ryanodine receptor, and phospholamban phosphorylation were altered, and the alterations are associated with changes in the contractile state of the myocardium during sepsis. So during sepsis, these proteins are changing, and that changes the way the heart contracts. The present study is dealing with sepsis-induced changes in the intracellular redistribution of DHPR, dihydropyridine receptors, represents part of the systematic investigation regarding roles of calcium regulatory proteins on the pathogenesis of altered cardiac function during the process of sepsis. Okay, so we're going to look at how the Voltsch-Bennett calcium channel changes during infection, sepsis. And the animal model, we're not going to read this, but CLP is the animal model, cecal ligation and puncture. So they tie off the large intestine and they puncture it, so the bacteria go out into the peritoneal cavity, and it causes this sepsis, which is basically bacteria in the blood, which is as bad as it gets. When you go septic, it's not good. Okay, results. So the first experiment here, figure 1, shows the time course of PN200-110 binding. So PN200-110 is a is a dihydropyridine. So they use, it's very high affinity dihydropyridine. So they're using this to test, to find those receptors, to do their binding studies to find out how the Bmax and how many receptors are there. Okay, so this is a time course. So you can see over a period of 60 minutes, it saturates, okay? Or it comes to equilibrium, I mean, not saturates. What's the difference between A and B? A is light vesicle prep, and B is sarcolemma on this side and light vesicle. All right, so Bezuit, do you remember what I said about sarcolemma versus light vesicle? What's the difference between those two?
In this experiment or in general?
Yeah, in this experiment. Why are they? I'm sorry, go ahead.
In this experiment, the sarcolemma has higher, no, the early sepsis is higher than the late sepsis.
Okay,
but when
wait. Don't worry about the conclusions of the study. Just what do they mean by sarcolemma versus light vesicle?
Sarcolemma is the membrane.
Right. And what's the light vesicle fraction?
That one I don't get.
Okay. Anyone else know? Anyone want to chime in here? Okay,
is it the vesicles in like inside the cell?
Right, exactly. So it's like I think I showed you the figure last time. It's like you've got your sarcolemma, and then underneath it there's always like a layer of vesicles there of proteins that can be quickly put into the membrane if they're needed, or proteins get taken out of the membrane and they go into this vesicle pool. So this is a pool of vesicles underneath that forms kind of like a buffer, like a pH buffer, so you can change things quickly. I don't know if the pH buffer metaphor works, but it's basically cell surface versus right underneath the cell surface in vesicles. All right, so who's next on the hit parade? Oh, Celine, you just answered that one. So Olivia, figure 1, why are they doing this experiment, do you think? They do explain it. What is it? What do we learn from figure 1?
Sorry, I'm kind of having trouble pulling up the article and I can't see the screen share.
Okay, let's move on to Matt Gamelin. What do we learn here?
We learn.
It's not a deep experiment. It's a pretty simple thing, really. See if it comes back up. Okay, where is it? Here's where we're going. This is the point of that experiment. So the bindings were saturable, which means it's specific. It's finding a receptor and binding to it. If it's non-specific, then the more drug you put in, the more binding you get. For both membrane fractions after 60 minutes. Based on this data, a 60-minute incubation time was adopted for the following experiments for the comparison between control and sepsis experiments. So basically, this is a control experiment, and controls are really important. So every other experiment after this, they're going to incubate the receptor and the drug together for 60 minutes. Now, if they didn't show this experiment, you'd be like, well, how do you know it's saturated? How do you know it isn't like somewhere else on that curve? So they show this to show you that, we're doing 60 minutes because we know at 60 minutes, the reaction's done. We've reached equilibrium, and then we get good data from it. Because if you can imagine, if you were doing your experiments and you're out in here, right? If you ended the experiment, a minute early or a minute late, it would make a big difference in how much is bound because you're in a very steep part of the curve. But out here at 60 minutes, it's done. So you don't have to worry about that so much. Okay, moving on. Now the next one is a really important one. Figures 2 and 3 show the effects of sepsis on the dynamics of dihydropyridine receptors in rat heart sarcolemma and light vesicle fractions based on PN2 and 110 binding studies. As shown in figures 2A and 2B, let's look at 2A and 2B. Oh, wait. The light vesicles exhibit a saturable process with the single component binding characteristics. So this is something that I talked about in the first lecture, but I didn't really hit it so hard. So I just want to explain it to you. So if you remember, I said if you've got a simple bimolecular reaction, like ligand binds to receptor, that's a single component binding. It's simple. And the slope of your binding curve is like between 10% and 90% is 2 log units, right? So if this was complicated, that slope could change. You know, if it's a cooperative binding, like one molecule binds and that helps other molecules to bind faster, then your slopes can be really steep. Or if it's uncooperative, it could make it more wide. But basically what they're saying here is it's a simple binding experiment, and that doesn't complicate our interpretation of the results. Okay, so here's our binding. Now, I talked about this Tuesday, I think, but I was a little bit disappointed that they didn't do nonlinear least squares. I like those better than these linear plots. But be that as it may, what are we up to? Ariana Totarum, you haven't said anything all semester. I'm really upset with you. What do we learn here in figure 2A? We can skip 2B.
For 2A, isn't it showing that early sepsis is increasing while the late sepsis is decreasing?
Right, exactly. So early in sepsis, the number of calcium channels at the surface goes up. Is this surface? Yes. What is this? Sarcolemmal membrane. This is sarcolemmal membrane.
Yeah.
Right, this is sarcolemmal. So early it goes up and late it goes down. And the scatter plot shows the same thing. It's just that you're looking at the x-intercept rather than the thing itself. Okay. And here we get numbers. So the maximal binding capacity, Bmax, for PM200-110 in sarcolemmal membrane fraction was increased by 27% in early sepsis, but decreased by 39% in late sepsis. The binding affinity, the reciprocal of the dissociation constant, for PN200-110 in sarcolemmal membranes remained unaffected. So what does that tell us? The fact that the KD didn't change during this experiment between early sepsis and late sepsis. We're up to Dante at this point. What do you think that means? Remember back to the first lecture, I was talking about KD versus Bmax. Bmax is the number of receptor sites in that tissue. What is KD all about? It's the affinity, but what determines affinity? Dante, bring your face up so I can see you struggle with this. Dante, are you sleeping? Oh, there he is. Okay. What do you think? You're still muted. Say something to me.
Doesn't it have to do with like cooperative binding or something like that? Like a strength binding?
Not really. What determines the affinity of the drug for the receptor? Going way back to the most basic. See, Dante, this is the point where you go into Spanish and we all just have to nod our head and agree because we don't speak Spanish. Oh yeah, you must be right. Yeah. Sounds authoritative. No, but seriously, what determines how that drug binds to the receptor?
The amount of the drug, no?
Part of it, yeah. But if you remember back to what the first lecture, I was saying that the shape of the drug molecule and the shape of the receptor determine that KD, right? So how well does this fit into this? So the fact that between early sepsis and late sepsis, that doesn't change, the KD is the same, tells you that the receptors themselves aren't changing, they're just more or less of them, okay? So it isn't like a conformational change is happening that is changing the binding, because that'll probably affect the Bmax too. It's just that there are more or fewer of them. So it's kind of like a control to show you that, you know, it is what we think it is. Okay, light vesicle fraction. Tyler Montano. Oh, see, he would have understood if you were speaking Spanish. So Tyler, maybe you don't. Tyler, do you speak Spanish? No. Do you? I don't know. Doesn't matter. Anyway, figure 3. This is the light vesicle fraction. What do we see here, Tyler? Tyler?
We see the effect of sepsis.
Uh-huh. What happens in early versus late?
In early versus late, we have, I don't know, I think we have higher efficacy in early versus late.
Bmax, not efficacy, Bmax.
Yeah, Bmax, high Bmax, yeah.
This is control. What happens from control to early? It goes down.
It decreases.
And then in late sepsis, it goes up.
Yeah.
Okay, so we're looking at the light vesicle fraction. So in early sepsis, it decreases, which suggests that they're going into the membrane surface, right? And late sepsis, it increases, meaning they're coming from the cell surface down into that light vesicle pool again. So it looks like we're watching just changes in the position of that dihydropyridine receptor between early and late sepsis. And again, the binding affinity is unaltered. Good. Similar results were obtained when PN200-110 bindings were expressed per gram weight myocardial tissue. So that's just about, the prep for making sarcolemmal vesicles versus the light vesicles. So just showing you that it's okay. It works out the same way. Because the Bmax for PN210 binding was increased in the sarcolemmal membranes, but decreased in the light vesicles during early phase of sepsis. Conversely, the Bmax was decreased in the sarcolemmal membranes, but increased in the light vesicles during late sepsis. It is suggested that dihydropyridine receptors in the rat heart were externalized from light vesicles to the sarcolemmal membranes during early sepsis, whereas they were internalized from the surface membranes to intracellular sites during late sepsis. It is of interest to note that the sum of the Bmax calculated based on gram weight of the myocardial tissue of sarcolemmal and light fractions was increased by 16% during early sepsis, while it was decreased by 17% during late sepsis. Okay, well, let's look at that one. What are they saying there? Bmax calculated on point of sarcolemmal. So here they're adding together the sarcolemmal and light vesicle Bmaxes and comparing that total amount of receptor before early sepsis and late sepsis. So what are we learning from that, Mike West?
I'm sorry, can you say that last little bit again?
Yeah, it's right here. Bmax, stop already. Go away. Bmax of sarcolemmal and light fraction vesicles was increased by 16% during early sepsis, while it was decreased by 17% during late sepsis. So basically, they're adding the Bmaxes of both the sarcolemma and the fraction together and looking at it in early versus late.
So contraction is stronger in early sepsis, and then the efficacy of it goes way down during late.
Right. And
basically what that's saying.
That's true, but that's not what this is saying, though. This is just looking at the number of calcium channels in early and late sepsis. So what are we seeing here?
So does the cell start to synthesize those calcium channels because they're getting destroyed, and then eventually just can't catch up, and they're all end up getting enough to kill the person?
Kind of. I don't know about the kill the person part, but you're right when you say the total number is increasing and then decreasing. So we're looking at two different things going on here. One is the movement from that internal pool out to the surface, and also the total number is increasing and decreasing. So, everything about the calcium channels is being changed in this sepsis model. Okay, that makes sense?
So only the amount of calcium channels increased? increase in the early and then decrease in the late.
Right. So the total amount increased early as well as being expressed more at the surface, and the total amount decreased as well as being pulled away from the surface.
Does this affect the DHPR?
That's what we're looking at, the DHPR. Yeah, the
Alright.
total number of these DHPRs.
Okay.
Okay. Okay. And as Mike says, that's going to affect the hemodynamics, right? You've got more calcium channels, it's going to contract stronger. You've got fewer, it's going to contract weaker. These data seem to imply that in addition to the intracellular redistribution, DHPRs were overexpressed during early phase, whereas they were underexpressed during the late phase. So if I just kept reading, we could have saved ourselves all of that difficulty. Okay. Figures 5 and 6 show the effects of sepsis on the photoaffinity labeling of dihydropyridine receptors. So remember, we talked about this the other day. You use that azidipine and you do your binding, and when you reach equilibrium, you zap it with light and it becomes covalent, and then you can run that on a gel and see where you can actually look at the protein itself. Instead of just looking at the binding characteristics, we're looking at the protein. So a major binding peptide with molecular mass of 165, and if you knew the literature, you'd know that the dihydropyridine receptor is 165 kilodaltons, so it's in a protein of the proper size. Was labeled in both membrane fractions of all 3 experimental groups after proteolysis. The covalent incorporation of zidipine into 165 was completely inhibited by nifedipine, which is a cold dihydropyridine. Okay, so what's that for? That's a control. What's that telling us? Celine. So this is a negative control. The covalent incorporation of azidipine into the 165 kilodalton peptides was completely inhibited by nifedipine, which is not labeled, a specific calcium channel antagonist.
So we're just talking about like how that part isn't being incorporated in like the data that we're looking at, right? Since it's being blocked, it's not supposed to be in there.
Mm-hmm. I'm not sure. I don't think I know what you're talking. I don't know what you're talking about.
Because like if it wasn't blocked, then it would be like interacting with the whole cell, and like we don't want that to react because that's not what we're looking at.
Right, right. So this is showing that it is specifically binding to the dihydropyridine receptor. It's not binding to some other weird thing. And we know the 165 is the dihydropyridine receptor because we know nifedipine is a dihydropyridine, or is a calcium channel antagonist. So let me say that again, maybe with a little more clarity. So the basic idea is you're putting this photoaffinity label into this gamish of, it's the sarcolemmal membrane. So you've got dihydropyridine receptors there, but you probably have some sodium potassium ATPase, all kinds of proteins there. So when you do this, you're labeling this 165 kilodalton protein. Good. How do we know it's actually the dihydropyridine receptor? Well, if you do the same experiment while competing with a really high concentration of a drug we know binds to the dihydropyridine receptor, and then we get no more labeling, that tells us that the labeling is to a dihydropyridine receptor. Okay? Does that make sense? Let me throw this one out to the crowd because this is kind of important. Let me see your faces. How many people understood that? Sort of. Dante Quinones, Matt Camillin. All right. Dante, yay, nay. Ariana, yay. Olivia, how you doing, Olivia? Did you pull up the paper yet?
Yes, I yeah.
Okay. So did everyone follow that explanation of the negative control here? Yes?
It's it.
Okay, good. So it's just showing that it's this binding is the photoaffinity labeling is specific for dihydropyridine receptor. Okay. These findings indicate that the 165 kilodalton peptides labeled with azitapine possess dihydropyridine specificity. Okay, so that's exactly what we were just talking about. Okay, as shown in figure 6, the binding for labeled peptides during early sepsis was increased by 28% in sarcolemmal membranes, while it was decreased by 23% in light vesicles. Okay, control early went up, light went down. The sum of the 165 labeled peptides in the two membrane fractions was increased by 13% during early sepsis. So when you add the two together, increases in early sepsis and decreased by 13% in late sepsis. So this reminds me of something. What does this remind me of? Oh, look it over here. So this is really important in terms of the way you write a paper. So they could have did a paper with just the binding data and it would have been it'd be an okay paper, but here they're showing by a totally different technique, they're getting the exact same results, okay? Here the total Bmax increased by 16 and then down by 17. Here the total protein they're measuring went up by 13 and down by 13. So it's about the same. So two different methods of looking at this peptide or the dihydropyridine receptor give you the same result. Also here, early sepsis up by 28, late sepsis down by 23. What did we see with the binding experiments? Early sepsis up by 23, late sepsis down by 39. So not exactly the same, but similar anyway. And that's kind of important in terms of when you write a paper, typically you like to show every piece of the puzzle. So you're not, there's not going to be a question about, how do you know this was right, kind of. What do you like to see, like kind of the best paper would be show changes in RNA, show changes in protein, show changes in binding. Because then you've got RNA, you've got protein, and you've got function. You've got all that pieced together. That makes the strongest paper. Okay. During late phase of sepsis, these data using tritiated azitapine as a photoaffinity label probe reinforced conclusions drawn from PN201 binding studies that the DHP receptors in the rat heart were externalized from light vesicles to the sarcolemmal membranes during early sepsis, whereas they were internalized to the surface during late sepsis. Okay. All right. So now there's this big table here, and I don't want to spend a lot of time on this table. This is basically a control. So these are all the proteins that may be there somewhere, and they just want to show that the vesicles they're using are what they say they are. So sodium potassium ATPase is a sarcolemmal protein. Calcium ATPase, I think it's sarcolemmal also. This is a mitochondrial ATPase, total protein dry weight. So I don't want to get into this, but this is just another control just to show you that when they say sarcolemmal vesicle and light fraction vesicle, it's not chunks of mitochondria that happen to get into their prep. It's just a control for that. Okay. All right, so let's read the discussion. In this study, we first established the two distinct cardiodynamic phases in the septic rat model, then isolated and purified sarcolemmal and light fraction vesicles, followed by the assay of DHPR. The experimental sepsis induced by CLP exhibited an initial hyper, followed by a progressive hypodynamic cardiac states, which are closely resembled to the biphasic changes seen in clinical sepsis. The procedures adapted for the isolation and purification of sarcolemmal and light vesicle fractions have been used extensively for studies of receptor translocation between the surface membranes and intracellular sites. So here they're citing other work that have used the same membrane preps to show this effect. The sarcolemmal membranes and light vesicle fractions were highly purified and minimally contaminated by other suborganelles based on marker enzyme studies. So that's what that big, up on the top of this page, that big not graph, table is talking about. Sarcolemmal membranes are highly purified and minimally contaminated with other organelles. The techniques used for the assay of dihydropyridine receptors have been used successfully for establishing structural and functional relation of DHPRs in the mitochondria. It is important to note that the use of two different approaches for DHPR assays, namely the PN210 and the azidipine binding studies, have yielded identical results. That is, the density of DHP receptors was increased in sarcolemmal membranes, but decreased in light vesicles during early, whereas the density of DHP was decreased in sarcolemmal, but increased in light vesicle during late. The total number of DHP receptors in the heart was increased early, but decreased during late phases of sepsis. These data, together with those on cardiodynamic changes demonstrate that DHPRs were externalized from light vesicles to sarcolemmal membranes during early hyperdynamic phase, but internalized from the surface to intracellular sites during the hypodynamic phase. Furthermore, DHPRs in the heart were overexpressed during early phase, whereas they were underexpressed during the late sepsis phase. The functional significance, okay, now this is where they say, that's what the paper was about, and they clearly showed it. So why does this matter? The functional significance of the altered expression of DHPR and its intracellular redistribution between the two subcellular compartments during different cardiodynamic phases of sepsis is apparent. Calcium entry through the sarcolemmal DHP participates in the development of actional potential, oops, there's a typo that got through, action potential. Remember the plateau phase of the action potential, and furthermore, it triggers the release of calcium from intracellular stores and thus initiates the contraction of cardiac muscles through interaction with myofibril fibers. Because of this positive inotropic action on the myocardium and overexpression externalization of DHPR from light vesicles to sarcolemmal membranes would augment cardiac muscle contractility, whereas an underexpression and internalization of DHPR from surface membranes to intracellular sites would decrease decrease myocardial contractility because the overexpression and externalization of DHP correlate with the hyperdynamic cardiac state, while the underexpression and internalization coincide with the hypodynamic state. It is conceivable that the overexpression, externalization, and the underexpression, internalization during early and late phases, respectively, of sepsis lead to the development of two distinct cardiodynamic states during progressive of sepsis. Okay, so they're kind of beating a dead horse here. They're saying, you know, because you've got more out there in early sepsis, that's going to help you get this stronger contraction, and vice versa. It is of interest to note that the sepsis-induced alterations in cardiac DHPR, as presented in the study, are identical to those in the beta adrenergic receptor, alpha adrenergic receptor, muscarinic cholinergic receptor, as previously reported by our laboratory. Since DHPRs, beta adrenergic receptors and alpha adrenergic receptors are positively, while muscarinic acetylcholine is negatively inotropic to mitochondrium, our findings suggest that the alterations in the DHPR, beta AR, alpha AR collectively contribute to the development of two distinct cardiodynamic states during the development of sepsis, during which muscarinic receptors represent an attempt to counterbalance the collective actions. Okay, so what they're saying here is they're referring back to their previous studies where they showed a bunch of other things were changing as well. Do you have time after? Oh, this is probably, sorry. So what they're saying is they've done previous studies where they showed similar things happening with the beta adrenergic receptor, which, as we saw Tuesday, is positively inotropic. The alpha adrenergic receptor, which frankly, I don't know what it does in the heart, so I can't help you there. While muscarinic receptor is negatively inotropic. The muscarinic receptor links to that GI and inhibits cyclic AMP. So a lot of things are changing here at the sarcolemma. Most of them are leading to a increased cardiodynamic state. The exact mechanism responsible for the intracellular translocation and altered expression of DHPR in the heart during different phases of sepsis is not well understood. That's a nice way of saying we don't know what the hell's going on. It's kind of, it's empirical. We can see it's happening. Why is it happening? We don't really know. It has to do with changes in genes and changes in cytoskeleton and all that stuff, but they haven't even looked at that. So they're saying it's not well understood. The density of the DHPR has been reported to be increased in myocardial hypertrophy, whereas it is found to be decreased in chronic heart failure and endotoxin-induced cardiac depression. So that endotoxin is really, that's what they're modeling with this cecal ligation and puncture experiment, endotoxin. There are two possibilities that could account for the observed alterations. One is the post-translational modification of receptor moieties through phosphorylation-dephosphorylation reactions, and the other is the transcriptional regulation on the receptor gene transcripts. Okay, so if you've had me before, you've heard about receptor regulation. I may talk about it next Tuesday, but that's what he's talking about here with the phosphorylation-dephosphorylation. Like, are the receptors getting phosphorylated and internalized, and then dephosphorylated and externalized? And then transcriptional regulation, are they up and down regulated at the gene level? Phosphorylation and dephosphorylation of receptor proteins is a common mechanism for receptor translocation between the cell surface and the intracellular compartment. Phosphorylation internalizes the receptor, whereas dephosphorylation reverses the internalization. We have reported previously that the externalization of beta-adrenergic receptor in the heart during early sepsis was coupled with a concomitant decrease in the receptor phosphorylation, while the internalization of the beta-adrenergic receptor during late sepsis was accompanied by a simultaneous increase in receptor phosphorylation. So we'll talk about this next week, where phosphorylation controls its position. Alternatively, changes to the receptor gene transcription have been reported to be underlying mechanisms leading to altered expression of the muscarinic receptor, blah, blah, blah. So in the end, most likely what they're saying is most likely what's causing this movement from the sarcolemma to the vesicles and back and forth is phosphorylation, dephosphorylation, which is a very fast mechanism. But they also showed that the total number of receptors is changing. So that's got to be at the transcriptional level. So something about the bacterial infection is causing transcriptional changes in these cells. Okay. Any questions? Comments? So usually what I like to do on an exam is I'll take one of these figures, copy it, and then ask you a question based on it. Basically interpret the figure. So if you have any questions about this, we can talk about it, but if not, that's about all I have for today. Okay? Any other questions? Everybody happy? Have we done enough? Have we justified our existence today? I think we have. Okay, I'm gonna let you go. I just want to tell Tyler, I'm gonna set up a Zoom and send that to you because he had some questions he wants to ask. So anybody else? Nothing? All right. See you later, guys.